mycorrhiza, particularly in the nursery, and then the potential legacy implications of getting your mycorrhiza right or wrong in the nursery, what that could do for forest performance. But the focus of this talk is really going to be more around the work that we've done with bacteria, and particularly beneficial bacteria within forest systems. So I've also credited one of my team, Sarah Addison, as sort of one of the authors of this talk, because she contributed a few slides and has really just done a mountain of work in this area. So that's why, yeah, she deserves a lot of recognition for the advances that we've made in this area. Righty, so just providing a bit of a background to the topics and the specific areas that I'll be talking about in this seminar. Well, firstly, we'll go back to this diagram that we have of sort of breaking the tree down all the way to the molecular building block level, up to the cellular level, all the way up to the macroforms, and finally the actual standing tree. And with a view of considering how within these different components or levels within the tree, what the bacteria do in forests. Well, bacteria are fundamental to the resources, to the availability of the resources that trees use. We talked about mycorrhiza as being critical to the uptake of those nutrients, but it's the soil bacteria that play a massive role in making those resources available in the first place. We're also going to touch a little bit on the opportunities around the role of bacteria as producers of phytohormones, basically making hormones that influence growth and the properties of the growth that is made. So the influence on cellular structure, what types of tissue is formed, and the properties of that tissue. And then probably I'll spend the most time talking around the opportunities to use beneficial soil bacteria to reduce the stress that the standing forest state experiences in order to protect the value that is created by those trees over the course of 30 years, but we only get to realize within about a 2-week window. So starting off with resources. So we'll use the nitrogen cycle as a bit of an exemplar because it's probably the one that we know best, frankly. So again, lots of interactions, lots of interchanges and movements of nitrogen through different forms within soil, lots of different potential fates for nitrogen that it can move into for loss as well. However, one of the key things to note is that beyond fertilizer inputs, the only way we're really getting more nitrogen into the soil is through the activity of nitrogen-fixing bacteria. So they're either living in association with plants such as legumes, or they're free living in soil. Now, they operate, well, these bacteria are generally called diazotrophic bacteria, and they have an enzyme called nitrogenase, which basically converts atmospheric nitrogen into, which is about 70 odd percent of the atmosphere, and they can convert that into ammonia. And that's basically taking a really stable, unavailable form of nitrogen and turning it into a really simple mineral form of the element, which can be taken up by plants really quickly. So it takes a lot of energy, though, to break that into bond. It uses it also needs specific conditions, specifically a low oxygen or a no oxygen environment. So a number of plants have found a way to incorporate or live with these diazotrophs, and everyone knows about clover pretty much. It's a classic example. The plant provides the bacteria with the habitat that it needs and the energy that it needs in the form of carbon, and the bacteria basically rewards the plant by providing it with a source of nitrogen. We can also have this activity going on with free-living bacteria in soil. Cyanobacteria are a particular example, but it is generally less productive in terms of the amount of nitrogen that's made. But overall, and particularly within forest systems where you're looking at not necessarily needing a huge amount of nitrogen, but you are needing some nitrogen drip-fed over a matter of decades, it actually is a really important source of nitrogen. So the other opportunity for bacteria to really play a big role in this process is through the decomposition processes, particularly mineralization processes within the overall sort of structure of decomposition. So if we consider that you've got a resource in a soil, nutrients or well, some form of nutrients in soil, that's taken up into plant biomass. So you're growing different components of biomass that eventually hit the ground again through mortality functions, and they get converted into organic matter. So that's basically really far more complicated forms of molecules holding those nutrients, but they're not readily available generally. But one of the we know that the process by which big chunks of plant are converted into much smaller organic matter happens through the activity of insects. Saprotrophic fungi are really involved in that. And there is a potential for some of that organic matter, some forms of more complex nutrients and carbon molecules to be directly absorbed by plant biomass. However, the vast majority of those nutrients return to the plant through mineralization. That organic matter is broken down, generally predominantly by soil bacteria, back into those really simple mineral forms that are available for plant uptake again. So that role of the plant bacteria, of the soil bacteria in that mineralization process is just fundamental to sustainable productivity in our forest systems. So what happens when we alter things, which we do pretty much all the time? We can alter things by harvesting trees, by changing the amount of material available for decomposers to work on. We can also alter things by adding our own nutrients through fertilizer inputs. We're putting nitrogen in this case, but in many other examples, we can be putting in phosphorus and boron and calcium and potassium. We're putting these things into the system, or alternatively through our harvesting, we're taking variable levels of that material away. So how sensitive are the bacteria that underpin these fundamental processes to those changes? So we've explored some of those things in some long-term trials where we've we've basically had a forest at the end of one rotation. We've harvested it in different ways. So a stem-only harvest, you can see at the top panel there. Basically, you take away the stem, but at the stump, you've taken off all the branches, needles. It's just all left on site. We have a whole tree treatment as well. It's not pictured in here, but just imagine you cut down the whole tree and you take the whole tree away, but you leave the forest floor intact. And then the other end of the spectrum from the stem-only harvest, you take the tree, you take everything, and you also take the forest floor. So you're basically leaving it at the bare mineral soil. So that's our sort of gradient of ways of looking at harvesting treatments. And what we found is some 17 years after the treatments on the left panel were applied, we were still seeing massive impacts on the rates of nitrogen mineralization. So if you can imagine that forest floor removal treatment, not only had we reduced massively the size of the nutrient pool at that site, the rate at which the soil bacteria were making nitrogen available was roughly a third of what it was under the stem-only treatment. So there was less to work with, and they were providing it more slowly. Now, by the time the site had reached its maturity, that second rotation had gone all the way through, so basically about 28 years after those pictures on the left, we went back in and we did some more measurements, and we found that nitrogen mineralization had basically recovered, but you still would have had a period of, well, at least 17 years, probably more, where you were not getting, where the soil bacteria were not functioning anywhere near optimum levels. They were not delivering for the trees. But we've actually seen much longer term effects from some of our treatments. So for example, at 3 of our sites, again, we were able to monitor for an entire rotation. We started adding fertilizer when they were planted. Then we basically went through with that rotation until the end of, until the rotation was mature about 28 years later. I think it was 27 years at Tarawera, but at Berwick and Waterhill it was 28 years later. Now at all of these sites, the fertilizer treatment stopped somewhere between somewhere around 15 years before harvest. But 15 years later, when we did our end of our rotation measurements, we saw enduring legacy effects, that the bacterial diversity was reduced in the plots where we added fertilizer. Okay, so what's the functional outcomes of this? Well, admittedly, the counterexample to that was at the Berwick site, but that had a strong history of fertilizer use, and the diversity was already plummeted at that site anyway. So at the 2 sites where the fertilizer use was probably had the biggest impact, they were also tended to be lower nutrient and higher diversity to start off with. So in terms of function, what this meant when we had a look at those Tarawera and Woodhill sites was that the reduction in diversity because of fertilizer addition meant that the bacterial community could use a much smaller range of carbon compounds. It could break down a much smaller range of carbon compounds, and the rate at which it could break down those different forms of carbon was also much slower. So again, the use of fertilizer in this case had reduced the ability of the soil bacterial community to use a wide range of substrates, to break them down and return them to their mineral form. And again, this has pretty significant implications for the fact that this has persisted for at least 15 years after fertilizer stopped. And indeed, it's present at the end of the rotation. If you did this treatment again and treated these trees the same way, it's reasonable to think that that capability would be reduced even further with each successive rotation if you kept adding fertilizer, if you managed it the same way. We also picked up another set of enduring changes, and this time was in the ability of the bacteria at the sites to fix nitrogen. So that nitrogenase enzyme, the one that does the heavy lifting and fixing nitrogen, that's basically associated with the nitrogen fixation gene NIFH. And so we measured at the end of the rotation the impact of fertilizer addition across a number of sites, and also the impact of organic matter removal. So again, for the fertilizer addition, this was about 15 years after fertilizer use stopped. And for the organic matter removal, this was about 28 years after those treatments had been applied. So obviously, the more fertilizer with fertilizer addition, the abundance of NIFH decreased. But interestingly enough, with increased organic matter removal, we saw the abundance of the gene increase. And this was acting essentially to buffer that loss of nutrients. And that makes sense because all of a sudden the ability to fix nitrogen is incredibly valuable when the pool of nitrogen has been savagely decreased. So that's what we were seeing there. But the converse when you added fertilizer, that the ability to fix nitrogen, which is expensive and quite costly, is not that worthwhile when nitrogen is available. However, after nitrogen fertilization stopped and nitrogen became more scarce at the site, that ability did not come back. So again, enduring changes. And so the potential to actually set yourself up in a negative where you're trying to get a short-term gain from fertilizer use, but your long-term position could actually end up being negative. So clear implications that we need to think about what management in our forests is doing to the bacterial functions that drive nutrient acquisition. And again, we've seen multiple sites where a short-term gain has created a long-term deficit that actually overwhelms the extent of that short-term gain. And there's a real problem with this though, that because generally, I mean, it's only really now that we're building monitoring of soil microbial activity into our forest quality metrics. Unless you're looking for this in particular, you're not going to pick it up. You're just going to see there was a good productivity, there was potentially a productivity response to my fertilizer use, and then it went away. But the actual long-term effects to be an overall negative. And unless you're understanding what's happening to those processes, it's going to be difficult to quantify that effect, to understand what's driving your productivity responses. So now moving on to looking at hormones. Sorry, we've just got someone with their mic on. I think it's Elms SS. Just getting quite a bit of noise there. Oh, there we go. Right. So moving on to hormones. So this is sort of the second area where we're looking at how we can exert control over the behavior of the tree, how it grows. So soil bacteria, simply put, make a lot of plant hormones. Cytokinins, indole acetic acid, stearoylic acid, abscisic acid, also a range of hormones involved in defense reactions as well. And they're basically able to exert a direct effect on growth by manipulating the behavior of the plants through the production of these hormones. So we started exploring IAA production by soil bacteria back in about 2007, working at a site on the Canterbury Plains. It's in the sort of the Burnham area. So it's quite close to Christchurch, where I'm based. And what we did is we had an established framework within the site that had been established, I think about, 13, 14 years prior to the samples that we started taking from the site. And specifically, we had a strong weed control effect, basically 100% weed control versus none, and a fertilizer treatment in there as well. And to go with this, to try and untangle what we might see, we also captured a range of tree productivity data. So what did we find? Well, firstly, we found that weed control had a very strong effect on IAA production by soil microbes. So this was measured as the ability of the soil bacteria within you could incubate them and see, well, how much IAA they produce over a certain time period. And we found that the samples that we took soil, from plots that have been where weed control had been used, were producing massive amounts of IAA. Now, we had to be very careful about this because some of the herbicide treatments, of course, involve the use of indole acetic acid equivalents or analogues. But the samples we took were about 6 years after the final herbicide application, and those products have a half-life of perhaps 4 months, 5 months in the soil. So we were quite confident that what we were getting was actually what the microbes were doing as a consequence of anything we were picking up. Now, we didn't see any response to fertilizer. So what we did, though, was try and find out, well, what are the relationships? So where we're seeing a lot of IAA production, how does that relate to productivity at the site? And what we found was that there was a reasonable kind of correlation between our IAA equivalent production and the production on a site-by-site basis. Now, the plot we saw here, I'm quite happy to admit that their R-squared isn't particularly high, but it's more the distribution of the points that sort of started to get my attention. That when you had low IAA equivalents, you could have high or low productivity, but when you had higher IAA equivalent production, you only tended to get high productivity. And this is a trend that we have seen repeated over and over and over again for a number of relationships between something a bacteria is doing and productivity at that site. Anyway, the details of this particular study are published. I'll just flash back. So that's in that paper there, a long time ago now. But we can talk about that in more detail if anyone wants to get in touch with me. So what happened next to the site? Well, obviously what we picked up was really interesting, and it would have been really good to untangle quite what was going on with the weed control effect. Why was it doing what it was doing? But then a dairy boom intervened on the Canterbury Plains, and about 9 months later, that's what the site looks like. And unfortunately, many other sites as well that we lost, basically, as the landowners decided to convert them to dairy and bowled over 15 or 16-year-old plantations. But that's what happens sometimes. And unfortunately, that's been a bit of a trend for most of our work in this sort of area for the last few years, for a wide range of reasons, some of them sort of relatively trivial and some of them actually quite tragic. We haven't been able to progress our hormone research framework much further. We've done a lot of small-scale tests like this one that I just showed now, so a little bit more with IAA, more with gibberellic acid, zeatin, and we have identified sensitivities in microbial production of these plant hormones and how management might affect them. But for the last few years, we just haven't been able to set up a high enough throughput system to do this across multiple trials in multiple locations. The testing capacity is simply, we haven't been able to put it all together despite trying through multiple avenues. But we do actually have that capability now. So this is something that we're looking to really push forward in the next few years. So in terms of the implications of this work, frankly, we don't know enough yet, but there are all sorts of opportunities. And one opportunity that is already exceedingly well developed within forestry is the use of gibberellic acids to induce seed production. And this picture that I stole from, I should have remembered the name of the outfit, should have written it down, but a seed orchard somewhere in Western Australia. And then that's basically what you're seeing. They're drilling the stem, injecting hormones that induces a particular behavior in the plant. And in this case, it's the production of seed. So it's already well established in some components of the forestry sector. We just need to pick up the pace in other parts of it. Now, stress tolerance and the work that we've been doing in this space. Well, as I sort of mentioned, there's a lot of forest out there. And every year, only a very small portion of it is actually was actually realized for economic gain. So right now, there is a lot of exposed forest that is going to be subjected to, we're not entirely sure. We have a lot of known risks, a lot of known diseases and things like wind throw and other events that could disrupt forest value. But there's a lot of uncertainties that could come with climate change. And indeed, well, from a human perspective, we've already seen what happens when a new pathogen sweeps through the world. What happens if, what is our ability to change our trees? And the problem is we don't really have much ability to change our trees to deal with a new pathogen, simply because they're out there and they're out there for a certain period of time, and we can't reset the genotype as can be done with annual crop plants, for example. So what can we do in the forest environment to make these trees more resilient to unknown threats? So one of the things that we've been working on for a long time now is a system involving ACC deaminase. Now, just to give you a bit of a background, when plants sense a stress, they put themselves in a survival mode, and they do this by producing a molecule called, I won't say the full name, it'll take half a year, but a molecule called ACC. Now, ACC is produced all the time in plants, but at very low levels. It's also very leaky. It leaks out of the roots into the soil, so it never really builds up in high concentrations in the plant until a stress event comes along. And then the plant upregulates the production of ACC massively. And all of a sudden, it can't leak out quickly enough. The concentration builds up in the plant. And then that triggers another set of genes which convert ACC into ethylene in massive quantities. And that triggers shutdown reactions. It basically puts the plant into a survival mode, and it does the plant quite a lot of damage in the process. And one of the reasons for that is that the side product from the conversion of ACC to ethylene is hydrogen cyanide. And again, that does a lot of tissue damage, so necrosis and all kinds of things. So that basically puts the plant in one of two states. It's either it's happy or it's panicking. Now, and as a consequence, one of the things that's often observed is that the plant actually does itself more damage from the shutdown process than the stress ever would. So if that stress is transient, or if it's something the plant could actually have dealt with, the plant's done itself more damage trying to protect itself from something. And that seems quite odd until you realize that plants can't migrate, they can't get out of the way of an impending stress, and they don't know how long that stress is going to last. So therefore, it's sort of become reasonable to assume the worst case, because if it was the worst case and you don't protect yourself, you won't survive. Now, soil bacteria, because they like happy photosynthesizing and productive plants, have developed a way to sort of manipulate this response. They can attenuate it. And that's done by bacteria that can produce the enzyme ACC deaminase. Now, I mentioned ACC was leaky. So as it's exuded by the roots, bacteria with ACC deaminase can break it down, and therefore through stoichiometry, more is exuded out of the roots and more is exuded out of the roots. And it actually buys a window of time when that ACC is building up within the plant tissue. It takes it considerably longer to reach the critical threshold, which triggers the ethylene and the shutdown reactions. So it's quite a neat system. It buys the plant essentially a window of time for the stress to either go away or for the plant to realize it can actually adapt to the stress. It isn't going to get worse. This is within its levels of physiological tolerance. But the beauty of it is that if that stress is real and if it persists, the plant can still shut down and still protect itself if it's needed. It doesn't shut down the system, it just delays it. Now, we've been exploring the opportunities to manipulate this process for probably about the last 15 years, and we've made some quite significant progress in this space. So going back to that work we did at the Burnham site, as well as looking at IAA, we did look at ACC deaminase activity, and we found that weed control increased ACC deaminase, but also fertilizer application, which had stopped quite a long time earlier at the site, also had a legacy effect of increasing ACC deaminase activity. Now, then we did a little bit of work. Well, does ACC deaminase activity correlate with tree performance? And we found that it did, but only in the plots that didn't receive fertilizer. So it was a pretty confusing outcome. And again, it's detailed in the publication there, but it does suggest there's an opportunity to do something with the sensitivity of the activity of ACC deaminase. So I did some work sort of expanding out, looking at ACC deaminase activity across multiple sites and trying to tie that into productivity. And again, we saw the return of this classic plot. So across these 10 odd sites, we saw that there were a number of sites that had low ACC deaminase activity, here basically measured as the production of the end product of the ACC deaminase reaction, which is alpha ketobutyrate. So we saw that sites which had low ACC deaminase activity could have high or low productivity, but the sites with the higher ACC deaminase activity only saw high productivity. So again, we just saw that same shape of the graph. And the thing that you have to be careful with this, though, is that ACC deaminase activity will only relate to productivity at sites where there is a stress, where the ACC deaminase will actually provide an advantage. We've found there are a number of sites where ACC deaminase activity is high, but that site is actually quite well served. The trees are not under stress by the physiological measurements we can undertake. So therefore, it's not really related to productivity. It's just something the bacteria are doing for another reason. And it's not providing any productivity benefit. But at sites like these, which, as you can see, the mean annual increment is pretty low, these are stressed sites, and that is generally where we see ACC deaminase activity providing a reasonable correlation with productivity at those sites. And then we switched to looking at longer term effects. So we transitioned from measuring activity to actually looking at the abundance of the gene, the bacterial gene for ACC deaminase, that's ACDS. And going back to that fertilizer treatment, we found a really long-term legacy effects, that where we had added fertilizer, we were seeing at the end of the rotation, we were still seeing gains in ACC deaminase gene copy numbers. And we also saw them for organic matter removal as well. So again, this is a legacy effect that was persisting for about 28 years. So at sites where we'd taken away more nutrients, under, say, for example, that FF treatment, the sites were more stressed, and the bacteria were responding to this by making the trees tolerate that stress to a greater extent. They were allowing them to stay more productive in a harsher environment. And 28 years was enough time for them to basically come to a persistent state in this, to persistently have had this increase in the bacterial community. So all of these to date had just been observations, post-hoc observations. No, we hadn't actually intentionally gone out to try and do stuff or to change the ACC deaminase gene abundance. So a few years ago, we decided it was time to give that a go. So we set up a trial in quite poor soil with some trees that were known to be with a particular clone of tree, a genotype that was known to be very sensitive to stress. And we did our best to try and increase ACC deaminase gene copy numbers. So back in June 2016, we added a reasonable, well, actually no, a smallish amount of fertilizer to the site, to half of the plots, sorry, to half of the pots in this trial, apologies. And we found almost immediately that we created a massive increase in the gene copy numbers for ACC deaminase. And this slowly started to dwindle. So by May 2017, we'd seen it decrease somewhat. And then by December 2017, null and void, no effect whatsoever. The effect had dissipated. But we were sort of reasonably clever enough to throw in a drought treatment at the start of spring in 2017. Now, we probably would have got stronger results if we'd done it way back in the spring of 2016, but we had what we had, so we tried it then. But we still got significant results. What we found is that the trees that had not had fertilizer, the control trees, as soon as we put that drought effect on, basically just stopped watering them, within 2 weeks, we saw a reduction in growth, and then that continued down to 6 weeks. But for the trees that had had the fertilizer treatment, yep, they certainly started from a stronger point, but they maintained their productivity after that 2-week period, and they only started to show negative effects in the 6-week drought measurement. So it just worked with that theory. We'd bought them a window of time where they could maintain their productivity, but then if that threat was real, they shut up shop and started protecting themselves. Now, some of the early work we did in this area also suggested that the selection of tree genotype is also essentially a management tool for altering this activity. So what we did is at another site on the Canterbury Plains, so water stressed, temperature stressed, a whole lot of different things. We looked at a number of different genotypes all within the same trial area, and we found that we could measure, despite these trees perhaps only being about 2 meters apart from each other, in the local area directly around the stem, we were finding significant differences in ACC deaminase activity. And then when we plotted that against the productivity in the trees, lo and behold, the trees with the lowest amount of ACC deaminase activity associated with them were also the smallest, whereas the trees with the greatest amount of ACC deaminase activity associated with them were the most productive. So that gave us another angle to try and work with. So again, as part of that pot trial, where we intentionally tested the effect of fertilizer, we also looked to explore how different genotypes were expressing their ability to recruit these bacteria, if they did have an ability to recruit these bacteria from within the same soil. So again, we measured this in terms of ACC deaminase gene copy numbers So when we started the experiment, there was a little bit of a bump for clone 3, but there was no significant differences. This was about a month after we planted them. But then by the end of the experiment in December 2017, we had a really quite strong gradient of these different clones and their ability to attract these bacteria or recruit the bacteria that have this capability. And then when we looked at the known properties of these clones, I've just sort of anonymized them here, we found that C2 was a genotype known for high tolerance to a range of conditions, whereas clone 4 was known for having virtually no tolerances whatsoever. So this was starting to align with what people see in the forest. So then, to explore that properly, we went out into the forest, into a site that we'd established which had about 12 different clones in large replicated blocks. And we found a massive, orders of magnitude difference in ACC deaminase activity underneath these different clones. All on the same site, all treated the same way, but just huge variation. And the neat thing that we found is that the top 3 clones were also our most productive and most tolerant clones. They were less damaged by Dothostroma. They had less problems with nutrient deficiency. They had less problems with drought. And they were also the trees that were much better at recruiting the bacteria that could be providing them with enhanced stress tolerance. Now that's a really neat discovery, all well and good, but we decided to try and put these things together. We knew that we had some capability to increase the ability of the activity of this bacteria, or this particular bacterial function. So we applied a stimulant to see what we could do. Could we upregulate, let's say that clone 4 has really good wood properties. You really want that tree to grow in more places than it can. Is there a way to make it more stress tolerant? So we found that in fact there was. So you can see for our low end, we were able to increase the activity of ACC deaminase associated with those trees. And when we did some measurements of physiological state, they were more stress tolerant. However, for our high end from clone 9 and A, B, and C, it caused ACC deaminase activity to plummet. So obviously there's something going on. There's some sort of critical threshold in terms of communication or recruitment. And for our clones that were already reasonably good at attracting these bacteria, we pushed something too far and the process broke. So we have no idea what's going on there, but it shows us that there is a threshold in there. There's a complex interaction that perhaps we can do more with. So overall, there's a huge opportunity here, we think, to improve the tolerance of the standing estate. And it's not just specific threats, because that ACC deaminase, well, that ACC to ethylene pathway is almost universal. I think there are only 2 plants where that isn't considered an important way in which that tree or that plant deals with stress. So this is something that's involved with a universal plant stress tolerance. So from what we saw, there's obviously opportunities to optimize what we do. We think we've got to start working with tree breeders to actually identify what is going on. Why are what is the actual signal that some of these trees are sending out that others aren't, to recruit these useful bacteria? And above all, we need longer term data to actually understand what the longitudinal effect of any changes on stress tolerance we make will be. Because we haven't looked at this for longer than about 3 or 4 years so far. At the outplanting site where that last data set came from, we have the opportunity to look at it over a rotation length, but we're right at the start of the rotation, so we're just going to keep monitoring that for a long time, I would say. Now, just going to close up now. So Yagani Islami has also been a really big part of this research, did a lot on that pot trial. So thanks go to her, but also, again, a big team from Scion, and also a lot of support from the New Zealand forestry sector, who have become quite enthusiastic about the potential that this seems to be offering. And again, thanks to Jody and the FWPA for this opportunity. So that's the end of that.
Great. Thank you very much, Simeon. Clearly there's a lot of potential, a lot of good discoveries you've made so far, but a lot of potential to do more work. Before we get into the questions, I'd just like to flag with people that if obviously there's clearly more work, so to gauge the direction and appetite for further research in this area in Australia, if people could email me, if you're interested in potentially participating in a workshop to develop where we want to go with this, you know, the potential to develop an investment plan for research in this area. So it'd be good to identify what capacity and capability we have here, and what are the priorities, and what's the appetite to do this type of work. So it'll, you know, it might be in the next couple of weeks, the next month or so. So please email me if you know of your colleagues who have been in on one or two of these webinars before. Pass that on to them as well if they're interested. Send me an email. And Simeon's offered to be as a part of that workshop as well. So, you know, it's a fantastic offer.
Yeah, depending on when it might happen, there'd be a couple of other people from Scion as well with expertise who have put their hand up and said that they could also sit in and help out as well. And they're probably more at that sort of project management, project development side of things.
Fantastic, thank you. Okay. Also, before we get into the questions, just to let you know that these webinars and Simeon's presentations will be uploaded onto the FWPA website by about Tuesday, so you can access them there if you want to review them, or if people have missed out on being able to attend them. So we will get into, we have a couple of questions here. Kevin Harding, what were the impacts on average stem size or productivity at Berwick, Tarawera and the other site? Of course, wearing a short-term forest manager's blinkers, so ignoring the potential for long-term impacts of 2 and 3R, running down soil microbe populations and their role in providing background nutrients.
So how we sort of looked at that sort of stuff was the relative trajectories and change in mean annual increment in the control and the fertilized plots through time. So obviously we got a bump in the fertilized plots, and then that went away, but then we could see it actually starting to dip below where, and this is only really true on the sites that were particularly nitrogen limited. At Berwick, we saw really nothing because that site was just full of nutrients. The fertilizer did virtually nothing anyway. But at the sites where nitrogen was limited, we saw a bump, but then their productivity dipped below the control plots at the same point in time. So you just basically can just do the differences of the relative differences. How much time do they spend above the curve? How much time do they spend below the curve relative to the controls? And we were seeing that you were ending up in about the same place, maybe slightly better, sometimes slightly worse, but you'd spent a bunch of money to basically spin your wheels in the end.
Follow-up question from, sorry, this is from Wolfgang Drexler. I'm interested if any work has been done on soil bacteria under burnt soil conditions, i.e. what happens after a fire and what could we do about it?
That's an interesting one. I think I mentioned a few weeks ago that there had been some really nice work done in Australia around mycorrhizal recovery post-fire and just how intense the fire needs to be to knock out mycorrhizal spore sources. I don't know if anything like the same level of work has been done for bacteria. Certainly it's not something we've done here. And not to say I've looked for it, but I don't think I've ever come across anything of that ilk around soil bacterial function. So no, I don't. There could be, but I'm not aware of any.
From Kevin again, Simeon, did you try to fit non-linear models to your ACC and other responses? Looks like a threshold level situation where the response is flat above the threshold level.
Yeah. When I got a bit better at statistics than I was then, I have done some sort of step analysis where there is you can basically say if this value was above this, you go down this pathway or that pathway. And yeah, you can say that there are some pretty important step changes where, there seem to be certain values of activity. But again, it's all quite site dependent because that ACC deaminase effect, it's also interactive with how stressful that site is in the first place. You can have high ACC deaminase activity, but if the site isn't stressed, it's just not going to matter. So yeah, but there are cleverer ways to do that analysis, that's for sure.
How is ACC deaminase stimulant applied?
Well, we've got a brute force way of doing it, which is just basically chuck on a bit of urea, but we have a far more efficient way of doing it now that I can't talk about, unfortunately, because there's a bit of commercial stuff going on in that space. But yeah, there are, you can, if you got the right thing, just ground-based application, or you can fling it out at a helicopter at really low concentrations. Yeah, we've actually got a number of ways of doing it now.
Okay. Regarding the effect of fertiliser in long-term diversity of bacteria and nitrogen fixation potential, what were the nutrients and rates applied to the sites? Could you see a different result if there were underlying deficiencies in trace elements or nutrients other than N? Also, were the sites involved deficient in any nutrients prior to fertiliser application?
Yeah. So that was the key thing. At the Berwick site, it was already highly fertile. It had, I think, 12 times as much soil nitrogen as the Woodhill site, for example. So what we were doing with our fertilizer treatments there were just a drop in the bucket compared to the nutrition at the site already. So the bacteria didn't really respond because we weren't really making a change to their environment. Whereas at the Woodhill site, it's a sand dune, it's got phosphorus, and that's about it. So the amount of nitrogen we were adding was a considerable proportion of the pool of nitrogen in that site. It really was changing their environment. It was making an enduring change, and that's where we were seeing our biggest responses, such as Tarawera. Yeah, the trace elements were interesting that you add that twist to it. We're just actually doing some work on that because obviously there's a lot of cofactors involved in bacterial enzyme function. And one of the areas that we're exploring is that we actually need to transition our fertilizer approaches to, instead of feeding the trees, we want to feed the bacteria with what they need, and then they'll look after the trees for us. So yeah, but again, you've got to build in, well, there could just be some overwhelming deficiencies. You just don't have enough nitrogen at that site. Nitrogen fixation will only get you so far. So therefore you do want to add more nitrogen to the site, or alternatively protect your organic matter as much as you can. Yeah, but we do have a good spread. So we have seen that is an important interaction with the responses that we've seen across, the last decade.
What's the current and future means of routine testing? Is broad-scale site testing a genuine reality? Is it consistent, accurate?
That is a very good question. How do we take the analytical sort of things that we're doing here from research scale and put them in an operational context? And again, that's something, well, the reason why we're having these seminars is because we were having a workshop, or we were intended to have a workshop basically to address those questions within the New Zealand forestry sector. But then, of course, because of COVID, that didn't happen. But then Jody got in touch with me and said, so when are you going to have that seminar? And then we got to talking, and this is one of the things, this is what came out of it. But we have, well, I don't want to get too much into the detail, but we're already developing, a number of our forestry companies are developing far more intense soil sampling for soil nutrition sampling strategies, and we're developing a whole lot of ways where they can be as efficient as possible in terms of using sort of different layers, conditioned Latin hypercubes sort of approaches, so that they can do the minimum amount of sampling to cover the most possible ground and get the most possible leverage from their effort, whatever scale they may want to do. And that's becoming quite successful. Companies are looking to apply that in quite well, but we're getting a lot of soil samples coming in to analyze, that's for sure, from a nutritional perspective. Now, what we're looking to do is dovetail soil microbial sampling with that. Again, it's expensive compared to nutrients, and it's certainly it's a novel approach. But what we're looking at is where you're getting oddball results, when you're getting things that don't align with what the nutrition at the site should be. That's an opportunity for us to get an understanding. We're seeing probably the biggest potential for this management is in the nurseries and with the mycorrhizal side of things, but in the forest, because we're getting these stimulants that can make these changes really rapidly within months, or actually within weeks, frankly, and they can be applied just as you would apply copper for Dithostroma, just as you would apply, a broadcast fertilizer for or boron. Some of the solutions we have already fit straight into forest management practices they are. In terms of understanding the microbes, the microbial activity, that's always going to be more expensive. Though that said, the costs of gene sequencing and community sequencing seem to always be going down. But there's also a point at which, well, how important is that information for a forest manager? It's more, here are things that we've found that as a general rule of thumb make the microbes do what you want more than others. If you've got oddities that could help explain it, but we're just thinking of how we could make this just as some rules of thumb and maybe some treatments that you can apply that can just fit into normal forest management practice.
Okay. Kevin's put a comment up there. Back in the early 1990s, North Carolina, assume that is, state breeding co-op put in a trial with the biotech group to look at drought hardiness in loblolly pine. They found a different set of active genes being switched on in year 1 to year 2, so a maturity effect. So quite complex gene response that will get the tree breeders excited, and much better tools to crack today than 25 years ago.
Oh yeah, I mean, in as much as the association with ACC deaminase does seem to align neatly, the trees will have their own physiological adaptations as well. I mean, increased, control over the stomata and better or worse nutrient use efficiency. The work we're doing with ACC deaminase and the ability of some genotypes to attract them and others just having no ability whatsoever, that's just going to be part of what they're doing. And I fully agree that there's going to be all kinds of signals and genes in there that we might never untangle, but again, it's just coming back to what are the rules of thumb that we can apply at a broad scale or manipulate at a broad scale.
Were the ACC differences amongst the clones linked to both growth rate and survival? Also, when the stimulant was applied, did that have a different effect on growth between the two groups of clones corresponding to ACC response to stimulant?
Yep, any tree that got the stimulant was bigger than the ones that didn't. There was no, even the genotypes when the ACC deaminase decreased, the stimulant has other positive effects. So literally, there wasn't a stimulant by genotype effect. The stimulant was just positive, but we saw it being not significantly, but somewhat more positive for the trees that didn't have that ability to attract those bacteria. That said, this was, I think, 18 months after the stimulant was applied. We've done some more applications since then, and the growth data out of that series will just keep running, and we'll just see where we get to. In terms of survival, it was a numerical but non-significant advantage in survival with the application of the stimulant, and again, no interaction with genotype. But again, give it time, we might see it come out to be significant, or I actually kind of hope it doesn't, because I do want trees to measure.
Okay, you mentioned that you can use urea to boost ACC activity. Does this mean that urea fertilizer can help reduce stress response in plantations?
Yeah, that one took me forever to figure out, because it seems a totally perverse response where you're adding nitrogen, which should not make the plant more stressed. It should make it happier. So therefore, my original thoughts, or my original hypothesis going back into that in that 2007 study, was that we'd actually see the other way around, that the ACC deaminase activity would be greater in the unfertilized plots. But consistently, everywhere we've gone, we've seen that fertilizer increases ACC deaminase activity. Well, yeah, across the board, we've seen ACC deaminase activity increased by fertilizer application. So the best pathway that I can see from understanding it is that the increased amount of fertilizer, or the increased amount of mineral fertilizer, turns on IAA, turns on a whole bunch of growth responses. The plant realizes it's in a better environment, it can grow more quickly. But one of the side issues with enhancing your IAA production is you can also lead to a cascade of effects which does increase ACC activity as well. I mean, and that's how herbicides work. They overstimulate IAA production to the point of the plant damaging itself because it turns on its ethylene pathway. So I don't know the answer to that one, but I'm thinking that that's what's happening. So again, we need to do some fairly deep biochemistry work and gene activation work to figure that one out. But I think that is our most likely pathway for why nitrogen, which should just make it a better environment, is triggering something in the bacteria to make them help reduce stress.
Okay, thank you so much. Thank you. It was an area of R&D that wasn't on my radar and looks like has huge potential. Also emphasises the importance of maintaining long-term trials and the value for new areas of R&D like this cannot be missed. Yes.
And I have to say that long-term trial thing is absolutely relevant. I did the first decade of work in this area was basically trials that were set up for something else, but they established gradients that we could go into after those treatments had had long enough to manifest a real effect on the environment, create a different environment, created a different soil microbiome, which that we can now see has quite different properties and capabilities, and that reflects in how they're interacting and how they are supporting or not supporting the tree.
Okay. Well, thank you. If there's no more questions, thank you so much, Simeon, for the presentation of these 3 webinars. It's been really good interest, and look forward to people contacting me, expressing interest in a workshop to further this thinking about where we go from here. Yeah, there's some few expressions of thanks from the listeners, so yeah, much appreciated, Simeon.
Cheers, no, good fun.
Yeah, thank you. We'll talk again soon. Thanks very much for everyone for your participation and questions and interest in this area.
Right. Cheers all.
Bye.
Thanks everyone.